to the second video PowerPoint in bacterial diversity, and this will walk you through the largest group of bacteria ever, and that's phylum Proteobacteria. Going back to the phylogenetic tree, just to remind everyone as to where we are, bacteria are almost the latest branch, if you will, highly diverged from the ancestor that is the common ancestor for all members of this domain. Bacteria, as represented by the size of this box, are also numerous and highly diverse and constitute a large and dominant phylum. So I've made this point a few times that it's large. Not only is it large, it's also highly metabolically diverse. So what that means is you find some phototrophs in here, some chemotrophs, and even among chemotrophs there's lots of diversity in terms of carbon source requirements as well as electron requirements. Based on purely 16S rDNA phylogeny, not based on nutritional status or energy or any of that, but just purely based on small subunit rDNA phylogeny, there are 5 classes of proteobacteria that are called alpha, proteobacteria, beta, and so on through epsilon. These classes themselves are so huge that they resemble phylum. And you will find that phototrophs are in more than one class, and you will find the chemolithotrophs and sulfobacteria being in more than one class. So there's lots of overlap between members in different classes. Within this broad group of phylum Proteobacteria are some chemoorganotrophs, including Pseudomonas. We will briefly look at these because these are ecologically important as well as medically important. Acetic acid bacteria again have huge ecological implications, so we will look at them. And let's not forget nitrogen fixers that are free living, extremely crucial for the ecosystem. But also in this group are pathogenic species like Enterococcus, Neisseria, Vibrio, some of which we will look at right away, and yet others we will revisit at the end of the semester. But besides these chemoorganotrophs, there are also what are non-chemoorganotrophs, for want of a better term. But in this broad umbrella are the phototrophs, and we've previously seen the bacteria, both purple sulfurs and purple non-sulfurs, that are part of this group. But also chemolithotrophs, things like iron bacteria, hydrogen bacteria, ammonia bacteria, some fascinating ways of life, and we will consider a couple of examples in this category. There is also bacteria that are called methylotrophic, which means they grow on methane or methyl group containing compounds. We won't look at them, but please understand that they fall within this phylum as well. Although this is not the first time we're considering this way of life being a chemolithotroph, methanogens after all are chemolithotrophs. These bacteria are worth looking at again because they're not exactly the same as methanogens, and also it's just so fascinating because this is the only domain where you could possibly encounter this metabolic way of life. So among chemolithotrophs are diverse groups that derive energy from oxidizing several different inorganic chemical compounds, and I mentioned nitrate, hydrogen, ammonia, hydrogen sulfide, etc. But among this broad group, there are a couple of genera that are incredibly dominant and are considered key genera, and that would be Nitrosomonas and Nitrobacter, both of which are crucial to the cycling of nitrogen in the ecosystem. Most chemolithotrophs also happen to be autotrophs, which means they're also capable of carbon dioxide fixation, and that's what methanogens were. They were also capable of CO2 fixation. But some chemolithotrophs are considered to be mixotrophs, which means they use two different sort of chemical compounds. Technically, the one from which they derive energy would be inorganic, and that's why they are lithotrophs. But they could still be using an organic chemical compound as a carbon source. So there is this sort of hybrid category that exists, but for the most part, we're going to be focusing on the pure chemolithoautotrophs, such as the hydrogen bacteria. So here is a hydrogen bacterium. An example is Ralstonia eutropha. So this is a specific example of hydrogen bacteria that are also proteobacteria and happen to be chemolithoautotrophs. I will remind everyone that what you need for this way of life is a way to produce energy in the form of ATP and a way to produce reducing power. In this case, it's in the form of NADH. By a combination of energy and reducing power, these bacteria are then able to fix carbon dioxide into the cell and incorporate it. So, in effect, they become autotrophic, but derive energy and reducing power from an inorganic compound, which is hydrogen in this case. How are they able to do this? Is by possessing some unique enzymes. The enzyme that immediately comes into play are called hydrogenases, and Ralstonia possesses two of them. One of these is a membrane integrated hydrogenase, so it's a membrane protein, of course, and it is able to split hydrogen and use electrons as part of an electron transport system. So hydrogenase passes on the electrons to quinone, which then passes it on to cytochrome B, C, cytochrome C, cytochrome A, and ultimately donates those electrons to oxygen, which accepts the electrons and forms water. Alright, but during these electron transfer steps, what you find is that discrete steps are associated with a huge negative delta G. And that means it can allow for the cell to do some work. And what work does it do? It sets up a proton gradient, which I hope all of you are familiar with by now. Proton gradient is set up so protons accumulate outside the membrane and can then flow back into the cell via the protein ATP synthetase, thereby allowing for the production or synthesis of ATP. So in a sense, we've taken hydrogen and made ATP. But also simultaneously, H2 being more electropositive than NAD can directly give up the electrons to NAD, allowing for the synthesis of NADH by the enzyme cytoplasmic hydrogenase. Thus, we've gotten both reducing power and energy with which to fix carbon dioxide. So, I mean, if that doesn't impress you, I know what else will. Here are bacteria that can just live on hydrogen basically and carbon dioxide, just like methanogens, right? But completely different way of going about producing reducing power and energy. So previously I described to you chemolithotrophs using hydrogen bacteria Ralstonia as an example, but also are present in this group what are called sulfur bacteria, and these are very different from our phototrophic sulfur bacteria, so let's not confuse them with one another. These sulfur bacteria are not phototrophic. They don't have any bacterial chlorophyll or any such thing, but also happen to rely on sulfur containing compounds for deriving electrons and may produce elemental sulfur as a byproduct, and that is displayed here for Beggiatoa. These tiny refractile granules are actually just sulfur granules that are accumulating, and they're also visible here in sulfur lobus, where a probe that specifically attaches to sulfur crystals, literally lights up the cells. These sulfur bacteria were originally characterized by Sergei Vinogradsky, one of the famous microbial ecologists. In fact, these are called Vinogradsky bacteria, Vinogradsky column, where he isolated these bacteria from. So Vinogradsky is a big name. Makes sense to shelve it at the back of your memory for now. So these sulfur bacteria are going to use some sulfur containing compound, whether it is hydrogen sulfide, or whether it is elemental sulfur, or whether it's thiosulfate, or even sulfite, in order to be gleaning electrons from them. After leaching electrons from these sulfur containing compounds, the usual byproduct ultimately is sulfate, which is the most oxidized form of sulfur. How they get there differs depending on what specific genus you are. Some of them use a system of enzymes called the SOX enzymes. The SOX enzyme is capable of directly going from hydrogen sulfide to sulfate. Of course, when I say directly, I mean the SOX enzymes are completely responsible for that. There's no intermediate byproduct, but it does happen in steps, and those steps result in deriving electrons which go through electron transport, and that electron transport will ultimately be connected to an energy production as well as reducing power production, similar to what we've seen before. But SOX system is not the only way to do this job. Yet other bacteria have the enzyme sulfite oxidase, which uses sulfite as the starting point and oxidizes it to sulfate, also generates electrons which go through the electron transport system. What about still starting with sulfite, but instead of using the sulfite oxidase, yet other chemolithotrophs use an intermediate called adenosine phosphosulfate, and to be able to do this they require a different enzyme which is called adenosine phosphosulfate reductase, and still produce sulfate as the ultimate oxidized product, and generate ATP by substrate level phosphorylation in this case. So various different ways. I don't want you to worry about the exact metabolic pathway here or the set of enzymes, but conceptually what I want you to get is that using sulfur is a way of life, not just for the phototrophic bacteria but also for these group like Bacterial, characterized by Vinogradsky, that are considered chemolithotrophs. We've looked at chemolithotrophs under proteobacteria, and now it's time to consider the other side of the coin. These are chemoorganotrophs, pseudomonids. Pseudomonids is a group name, but within this, a prominent member of this group is Pseudomonas. Pseudomonas, Zymomonas, Burkholderia, Xanthomonas, all of these group as pseudomonids, and a couple of features just unify them: the high nutritional diversity. Earlier I had mentioned at the very start of this chapter how Burkholderia can grow in as many as 50 different carbon sources, and that's the extent of diversity we're looking at within this group. Pseudomonas is capable of degrading aromatic hydrocarbons, and can grow on plastic, can grow on oil spills, and has therefore huge environmental application and biotechnological uses. Many members of this group are pigmented, and the pigments are called pyoverdin and pyocyanin. Pyoverdin is sort of verde green, so it looks a little green. This one is slightly blue green. So many of these bacteria have yellowish green sort of fluorescent color. Well, not truly fluorescent, but they do look bright. Most of them are motile, and you can see that with the polar flagella. And they're also rod shaped, and I said they have chemoorganotrophic metabolism, and most are capable of respiratory metabolism and require either oxygen strictly or grow better when oxygen is present. Besides the environmental applications for this group, Pseudomonas aeruginosa, one particular species of Pseudomonas, is a pathogen, and is considered to be what is called an opportunistic pathogen. Opportunistic pathogens are those that do not affect healthy individuals but are capable of causing infection only in immunocompromised patients. So they have some immune system trouble or stress or a previous infection that lowers the immune response, some situation that this bacterium takes advantage of. Such bacteria are called opportunistic pathogens, and one of them, Pseudomonas aeruginosa. It is frequently associated with respiratory infections in HIV positive individuals who have a lowered immune response. HIV, after all, causes immunodeficiency. Pseudomonas aeruginosa is also frequently seen in CF, cystic fibrosis patients who have trouble clearing the mucus in their body, and therefore have a lowered resistance, especially in the mucosal tissues. Pseudomonas aeruginosa is also considered what is called a nosocomial pathogen. Nosocomial is a term we refer to those pathogens that are acquired in the hospital setting. Pseudomonas aeruginosa is frequently that opportunistic and also nosocomial in nature. So in proteobacteria, we've looked at chemolithotrophs and pseudomonads, and that brings us to another group, the acidic acid bacteria. These bacteria produce acidic acid as a byproduct of their metabolism and grow fermentatively, which is why they're producing a byproduct, and take organic compounds and derive energy electrons from them, and then produce this fermentation byproduct, which is acidic acid. And because they keep producing acid, they're able to tolerate low pH and high concentration of acid in the medium. Example of this group is Acidobacter acidi, which is capable of using ethanol as a carbon source and produces acidic acid. And you can see that clearance around the colonies on the agar plate, that's due to acidic acid production, which literally clears up the opacity of the medium right around where the colonies are growing. Yet another group of proteobacteria are free-living nitrogen fixers, and these are aerobic. These free-living nitrogen fixers are actually widely distributed within proteobacteria in 3 different classes: alpha, beta, and gamma. These were discovered by Martinus Beijerinck, another very famous microbiologist. An example of the free-living nitrogen fixers is a bacterium called Azotobacter vinelandi, which is capable of existing as actively growing cells called vegetative cells, and that's what is pictured here. But under unfavorable conditions, Azotobacter also forms cysts. Cysts are the equivalent of endospores in some ways, but not in others. Or akineids in cyanobacteria in some ways. Cysts are dormant structures, resting structures, so they're not actively growing and reproducing or even metabolically active. And it is resistant to starvation and radiation, or nutrient depletion and radiation, but not to heat. So you can see how these resting structures in various groups have some similarities and also some differences. So the objective of nitrogen fixation is to take atmospheric nitrogen and fix it in the ecosystem in a usable form, typically in the form of ammonia, which is incorporated along with glutamic acid to make glutamine. That's the objective of nitrogen fixation, and Azotobacter is able to do that. Recall that nitrogen fixation requires the key enzyme nitrogenase, which is extremely sensitive to the presence of oxygen. But I just mentioned to you that these are aerobic in nature, so they require oxygen, but they need to protect their nitrogenase from oxygen. To be able to have the cake as well as eat it, and to protect the nitrogenase, Azotobacter has a thick layer of slime and capsule that surrounds their cell, and that capsule is apparent here. You can see the dense dark squiggle. That's the bacterium. What's outside is that slime layer or thick capsule, also seen in these electron micrographs as that thick layer outside. The colonies, thanks to that gooey carbohydrate slime layer, look shiny and glistening almost, all evidence of a slime layer or capsule present. So what if a slime layer or capsule is present? See, that slows down the diffusion of oxygen, just like you saw with the heterosis of cyanobacteria. They had this thick envelope of glycolipid outside, and here instead of glycolipid you have polysaccharide, a gooey carbohydrate layer which protects the nitrogenase from oxygen. So several adaptations, all with the same end goal, which is to protect nitrogenase from oxygen. The process of nitrogen fixation, whether or not you are a cyanobacterium, whether or not you are Azotobacter, or looking at rhizobium, which lives in root nodules of leguminous plants, it doesn't matter what kind of nitrogen fixer you are, but they all use nitrogenase. And nitrogenase is actually made up of two enzyme complexes. So there are two different enzymes that constitute the nitrogenase enzyme complex. These two enzymes are called DNR, dinitrogenase reductase, or dinitrogenase. So there are two of them with discrete functions that constitute this enzyme complex. With the help of this enzyme nitrogenase, nitrogen gas, which is N triple bond N, shown right here, is progressively going to be reduced and converted to ammonia. Reduction requires, of course, electrons. Electrons are provided to the system in the form of hydrogen atoms, and that's done in a stepwise manner. The series of reduction reactions requires considerable energy. So just to go from N2 to NH3, it requires somewhere in the range of 16 to 24 ATP molecules. It's a huge burden on this organism. Of course, that is outweighed or compensated by the fact that it gets access to hydrogen directly from the atmosphere, so it's a good price to pay. Pyruvate in the organism or some other organic compound becomes the source of electrons. Those electrons are passed on to the nitrogenase complex through a series of electron transfers. From the time the electron gets to the nitrogenase, from the time the electrons get to the nitrogenase enzyme complex, it's dinitrogenase reductase or DNR that first receives the electrons and undergoes an oxidation reduction cycle and passes on those electrons to dinitrogenase, which then accepts those electrons, becomes reduced, and then passes on those electrons ultimately to nitrogen. It's very simplistically represented here, but keep in mind that we can't go from nitrogen to ammonia in just one step. It requires a series of reductions, so every cycle requires a set of electrons to be pumped into the system. Phylum Proteobacteria also includes members of the genus Neisseria, which are diplococci, meaning they have a pair of cells that are stuck together, and that morphology should be apparent in this electron micrograph here. Two prominent members of the Neisseria group are Neisseria gonorrhoeae and Neisseria meningitidis, both of which are pathogenic to mammals. Neisseria gonorrhoeae causes gonorrhea, meningitidis causes meningitis, and a feature of both of them, a shared feature, is that they express type 4 pi that is very important in mediating adherence to host cells. We will look at Neisseria gonorrhoeae in considerable detail later on in the semester, but for now, please note that they map to Proteobacteria. Also part of Proteobacteria are enteric bacteria, which means these are bacteria that live in intestines of mammals typically, and several bacteria fit this bill. Some that you might have heard of are E. coli, Salmonella, Klebsiella, Shigella, Serratia, Erwinia. All of them are considered to be enteric bacteria or enterobacteria. Shared features would include the fact that they're all facultative aerobes or facultative anaerobes, both of which means the same thing, and therefore can grow in conditions that lack oxygen by fermenting sugars. But of course they would grow better by using a respiratory metabolism in the presence of oxygen. My battery is running low, but we're almost done. This concludes what I wanted to convey to you about bacterial diversity, specifically about bacteria. And then on Monday, what we will do is briefly go over, I won't reteach anything, but we will summarize and make sure everyone got what they're supposed to from these two video lectures, and then start with a new module which looks at bacterial genomes and chromosomes. Thank you.